can be done on Tuesday, just kind of as a refresher. And then your quiz is only on making cell suspensions and being able to tell the difference between a positive and a negative. So it's not, horrendous, but we'll review that anyway. You have a comprehensive lab practical, which was those 2 flow sheets I gave you yesterday. Were you able to find the other one? You didn't-
It's in the syllabus.
Oh, okay, good, all right. So again, comprehensive lab practical. Your practical, how many of you are there, 12? I don't know if we'll divide you in half or we'll divide you in thirds like we did chemistry, we'll see. But I give you 3 hours and to do the lab practical. And the fastest I've ever had anybody take it was 1 hour and 45 minutes, and then some people just never finish. So however you want to get your work done and get it going. But most people, I'd say, do finish it. So 2 presentations, I talked about this yesterday, 2 other blood groups and components. So next Monday, I'll go ahead and assign the other blood groups because it'll be due the week after that. Okay, and again, it's just a sheet that I give you, and then you just fill in the information. It's not a big deal. And then components is the components that you make from a blood donation, and that's like way at the very end. Okay. Again, 1 article, and again, it can either be an article or a current event, something you find in the newspaper, something you hear on the news, just something that you're aware of in blood banking. Just write up a couple paragraphs, just so the college knows that you can read and write in English. That's all I need. So, there will be some homework, but it's mainly panels when we get to panels, and then I do have extra credit if you so desire. So again, I told you yesterday that your ABO had to be with 100%. Now that is performing it, not your written test, but being able to perform ABO is 100%. From day 1 all the way through your practical. So if you get your practical all done, but you miss 1 ABO, it's still a failed practical, okay? So recognize and understand other blood groups. There's a lot of other blood groups, and we're going to talk about them. You're not going to kill anybody if you give them the wrong one there. You're just going to sensitize them, which means the next person that gives it to them might kill them, but it's not going to be you. So identify and detect and identify antibodies, perform accurate compatibility testing. That's really what I need to get you to. Jean's probably told you in micro, you never get to an end. You just teach. In blood bank, it's like writing a story. First, you learn how to spell, then you make sentences, then paragraphs, and when you're all done, you've got a story. Okay, my goal is to get you to where you can take blood from a patient who's ill and give them donated blood and make sure that you don't make them sicker. Okay, that's our goal. But if for some reason we do goof a little bit, then I gotta show you how to make sure you correct whatever it was that you did wrong and you don't kill the guy in the end. So transfusion reactions. And then a little bit of donor processing. Has anybody ever donated a unit of blood? So we'll go a little bit over the donor processing and the things that they require when you donate. And then component processing, but that's way towards the end. Quality issues in blood bank. You have to keep good records. You got to make sure that you know what you're doing. In blood bank, we have traceability and tractability. If we have a unit of blood that was donated and we gave it to somebody, we need to keep records for that person's entire life, okay? We keep them for 99 plus years, and finally somebody says, oh, I think this person be 110 today, and I think they're probably dead. So we can throw away their records, but we keep them forever. Back in the day, when I was a young tech, we had everything on little note cards, and they were in file cabinets, okay? Now with the computer world, it's a whole lot better, so you'll just look back and whatever. And if the patient has anything now, it kind of flashes at you and lets you know that there's a problem. So that makes it a little bit easier for you guys, okay? But if we give blood to somebody and then 5 years down the road, the donor comes up with HIV positive, all right, they're going to trace back to every time that person has donated. And when they donated, who got their specimen. So you have to be able to keep track and keep records of every specimen you give and who got it and where did it go. Okay, again, sometimes they expire and we throw them in the trash. You have to have that mark that we know where that is. Okay, we had a case just a couple weeks ago where we had an emergency donation, emergency release, and the tech forgot to go in the computer and release it, emergency release. And so here was a month down the road and they're like in a panic because the computer still says we have these units and we don't have these units. So at least it was been just a month and we were able to trace it down. But can you imagine if that was 3 years ago? Okay, so, and they keep those records forever. Standard operating procedures, again, you just got to be able to know what you're doing and do it right every single time. And then again, you have a quality control program. We will do a little bit of quality control with our reagents, but again, my reagents are old and I just hope for the best that we get something, okay? But when you get into the real lab, you guys know this because you were just there, right? What's the first thing you did in the morning? Your QC, right? To make sure your reagents and everything you were doing. So blood bank is no different. You have your reagents, you have your QC for the day. So it won't be any different there. Personnel qualifications. So again, we have to know who you are and do you have the ability to do what you're doing. So again, training new employees in standard operating procedures. Can they demonstrate the knowledge of what they're doing? Continuing education requirements. So you guys are the young group. I'm old. I don't have to do continuing education. I do. College still appreciates it, but nothing I have to document, and I'm not going to get fired if I don't do it. But you guys will. You'll lose your thing. My thing never expires. I think you guys, it's, yeah. Anyway. And then competence assessment. Did you guys do any proficiency testing or any surveys or anything while you were there? Did they involve you in some of those? Yeah, so blood bank gets them as well. Did you guys do it where one person would do it and then another person did it to make sure they matched it and then? No? No? Oh, just one person? That was pretty good. That's how you're supposed to do it. But usually we have 10 people that look at it before it goes out. Quality control, again, do it on your reagents. Make sure your reagents are working. Proficiency testing we just talked about. Standard operating procedures. It's, sorry, I guess I went one slide faster than you. So in your book on page 323, there's a box there that kind of goes through all that good stuff. You can read through there. Nothing too crazy. You've probably heard about it all before. It is important in blood bank that we validate. Well, actually with anything. Did you guys do any validation when you were there? Did you change any kind of a procedure? Maybe a kit or a reagent or anything where you had to validate to make sure that was working? They probably didn't maybe get you guys involved in that. With blood bank, it's important as well. And we'll get into this a little bit more when we start talking about an electronic cross-match. Because you guys are in the age where we trust computers, okay? And if we put the results in the computer and the computer says it's okay, we can release it without actually mixing the two blood specimens together, okay? So, but again, there's certain validations you have to do. Not only do you have to put it in correctly and the computer says, okay, it's okay, you can give this person a unit of blood, you also have to put it in incorrectly to make sure that the computer catches it and says it's not okay to give this person blood. So you have to do it both ways, okay? And it's done annually. But we'll talk a little bit more about that when we get there. Accuracy, getting it right every time, right? Close. Because I've been doing blood bank for so long, and plus I'm making up the stuff, and so I know what the answers are. I know how careful I have to look for the reaction, okay? So it's not fair to you to have to compete against me. So what I do in blood bank is I give 2 students, sometimes more than 2, the exact same thing, all right? So that you are being graded against the other student. If both of you miss it, I'm not going to mark you wrong because neither one of you caught it, okay? If one of you got it and the other one didn't, then we'll look at what happened and then grade you accordingly. So it's not that Barb knows the answer and you have to get the exact answer Barb gave. Okay, because sometimes it doesn't work out that way. Last year, I had 2 students, and it was their practice. They had not missed an antibody the whole time. It was their practical, and they were lab partners, and the first student missed it, okay, on their practical. And he threw a hissy fit. And I'm like, just chill, got to wait and see. If the other student gets it, well, then it's a problem. But if the other student doesn't get it, then obviously I didn't make it strong enough for you to pick up. And it just... luck of the draw, his lab partner was actually the one that came in next and actually picked the exact same sample. Again, he didn't know, and he missed it too. So therefore, I either didn't make it up strong enough, or it, for whatever reason, didn't work for whatever reagents they were using in their box at the time. So again, it didn't count against either one of them. So again, just to be fair. We are going to do some calibration with our centrifuge tomorrow, or on Friday, I'm sorry. I want to, not all the centrifuges are going to give you the same reaction based on how much time you put them on. So we're going to make a dilution of some antisera, and then we're going to make our suspensions, and we're going to see how long it takes for our particular centrifuge to get us a 1+, 2+, 3+ kind of reaction. And that way your centrifuge will be independent of anybody else's. So again, if we do this and you know that to get a 3+ reaction, your centrifuge takes 30 seconds where other people took 20 or something, I'll just ask that you put a note on it or mark it with a Sharpie marker or something so that if somebody else sits in your desk, then they know that for a 3+ reaction, you need to spin it for 30 seconds or whatever. Okay, but we'll do that on Friday. Regulatory agencies, we're going to actually talk about this a little bit more in the next PowerPoint, but those are them. So let's go on to the next one. So safety issues that we're worried about in blood bank. Regulatory agencies, people that are going to come in and inspect your laboratory, okay, for blood bank. The Food and Drug Administration. And why would the FDA care about your blood bank? What does the FDA usually come take a look at? Is it your reagents? What do they normally look at? They come to inspect restaurants, right, to make sure your food is okay. They're working in a factory that's producing a food product. They're going to come in and take a look and make sure you're keeping things sanitary, right? That's exactly right. Because a blood product that is being transfused to a person is considered a food product. So the FDA will come into your laboratory and inspect. And what are they looking for? That your refrigerators are clean, that you're not keeping anything else in them like you're not supposed to, right? Not keeping your lunch next to your units of blood. That you monitor your temperatures, so you have temperature charts. Do you remember that at all? Did you look at any of those where they actually had temperature charts where they're keeping track of your temperatures and everything else? And to me, it is just that magical thing that happens to a blood specimen that's a biohazard when it's in the lab. But the minute we put it in a clear plastic bag and walk outside the lab, voila, all of a sudden it's not a biohazard anymore and we can transfuse it to somebody. So it's that magical pixie dust at the entrance, I guess, that turns it from a biohazard to a magical food product. But anyway, the Food and Drug Administration will come in. The FDA actually in November passed a new ruling that they can actually come into workplaces and examine your workplace refrigerator, your break room refrigerator. Now, I don't know that they're just going to randomly walk into some place and check your break room refrigerator. However, if they're in the lab looking at your blood bank refrigerator, they could happen to actually go into your. So what they're saying is that you have to have documentation that your refrigerator is cleaned at least once a month. So if they go in there and they find, did you guys have to do that? Date your stuff? Or we have a sign on our refrigerator that says you have to date it and make sure that everything's fresh. But I mean, we have a couple people that are in the lab that just hate and clean out the refrigerator every couple weeks anyway, so I think we're okay. But anyway, that was a new ruling. OSHA, of course, OSHA, safety everywhere, right? So OSHA's going to come in and examine things too. The CDC, which again for standard precautions, where everything and anything is a biohazard. And also the United Postal Service, because we ship specimens through the postal system. Okay, so again, regulatory agencies, so they want to come in and make sure that we're packaging it correctly. I've never once had a postman in my lab, but again, I guess they could, to make sure that we're filling everything correctly and we're sealing everything like we should and making sure that nothing is going to leak. So they can actually come in and inspect us as well. Voluntary compliance, JACO. Did any of you have a JACO inspection while you were there? No? Did they talk about JACO at all? You did, because they came in October. Yeah, so a JACO came in when you did. CAP, so not everybody's CAP, but I know Breeze were trying to get there. So did you hear about that at all at Breeze? That they're trying to, yeah, because they came and pricked my brain to see how I answered the standards for safety. So I know they were trying to get their stuff up and running for CAP. I think Dr. Hagee's the one that's pushing that. But anyway, and then another one, yes.
So at Memorial, they told me the CAP was coming in, but then when they got there, they said that it wasn't CAP. It was people from another hospital coming to review. It was like a self-review.
Self-inspection.
But it wasn't from another, it was from another.
And we do that as a courtesy. We do that for other labs as a
What
courtesy.
is that called? They had somebody from state observing the people doing the reviewing to make sure that they were reviewing correctly, but
So every other year, you were actually inspected. And then on the off years, you have to turn in a self-inspection, which means you basically are grading yourself to see if you are matching the standards. Some labs choose to have another entity, and we have done that as well. So St. Elizabeth's has gone to Highland, and we have done a mock inspection for them just to see if, as inspectors, do we see anything that the real inspectors are going to come find, anything that's really glaring. So that may have been what you
like a
had.
local hospital, though. This was a hospital from like 4 hours away.
I don't know. I don't know if because it's BJC, it's something that is that you were at Memorial, right? So I don't know if it was a BJC thing and that was just something they chose to do. I don't know. Yeah, we had one come down from Chicago.
But that wasn't a cap inspection, was it?
I don't know. I don't know if it was CAP or what it was. We had a mock OSHA inspection too, and we had a mock. No, the EPA one was real, but the OSHA one was mock. So who knows? I don't know who it was that actually came, but it could be a BJC thing just because I know Memorial was just crossing over to BJC, and maybe they were coming to make sure that they were up to snuff before they actually. Yeah, I couldn't be. Diane might be the one to tell you. I really don't know.
Farms, they hire people to come in to inspect before they will inspection.
Right. Yeah. Let's see. You will hear me say AABB because, again, blood banks are also accredited by the American Association of Blood Banks. So AABB. So I will probably say something like it is an AABB standard that we have to label them this way, or it's an AABB standard that we have to do this. So you'll hear me say that every once in a while. And it's not because it's CAP or anybody else. It's the blood bank. Yes? You have a question?
The national database for all the bloods so that you can know if you had an issue like over at this hospital 10 hours away versus
We're
next
getting
one.
better, but there's a lot that's just individual hospital.
Is that because of HIPAA laws?
No, not so much HIPAA. I think it's just the technology just quite hasn't gotten there. But because of with the Sudafed and all that, and people buying it in different states, and so they're like, okay, wait, we need a national.
Right.
So I think they're getting there. We've had a pretty good, I would honestly say we have a pretty good rapport hospital about patients. If a patient is transferred to your hospital and you can call back and say, does this patient have anything? Or you'll ask the family, has this patient ever received blood from another facility? And you call to that other facility and say, did they have anything? So a lot of places, that's what we're doing more and more. But we're getting better about it. Yeah, but there's no national standard, which would be nice, but there isn't one. So again, AABB, you'll just hear me say that every once in a while. It's because an AABB standard. All right, I don't know what that is. All right. Great. So your policy will include, or your safety program should include policies and procedures. I'm sure you guys probably had to, well, if you got hired, you had to sit down and read them. If you, as a student, you probably didn't have to. Again, requirements and everything for whoever's working in your blood bank. Also part of the blood bank standard or process in blood bank safety, it's the only department in the laboratory that is required to have an exact physical space. So in other words, your blood bank shouldn't just be in a closet if you're doing X amount of blood work. Okay, so it's kind of based on how much work you do, how big your laboratory space actually has to be. Okay, so that's kind of a physical space. None of the other ones have that standard. Laboratory attire, again, you probably all got beaten over the head with PPE, right? Wearing your goggles and your lab coat and your gloves and all that good stuff. Biological safety equipment, you probably went to hoods for certain things and all that good stuff, right? So I'm sure as students, they probably wanted to show you the right thing and not the wrong thing. Did you notice a lot of people not wearing gloves? Did you notice people not wearing PPE? And when did the glasses come out?
I don't think they had glasses.
Really? When did the glasses come out?
When
When the CAP inspection happened and when
Basically.
Jaco was in the hospital, because we overhouse, right? Let's welcome Jaco to the hospital. Yeah, so anyways, that's usually when the glasses come out. But anyway. And again, warning signs and labels, so biohazard signs. You didn't eat in a biohazard room, right? And all that good stuff. So just again, warning signs. Standard precautions, you treat every specimen as if it's a potential hazard. Nothing strange or different there, right? Okay. And then cleaning and blood bank. So just as a requirement in all the other departments, you probably had to clean every shift, right? You had to spray it down with bleach water. Blood bank is no different. We do not bleach blood bags, so we'll actually get to that when we get to components, because blood bags actually breathe, and so if you bleach the outside of a bag, the bleach can actually leach into the bag. So you do have to be careful that you don't do anything around blood, but we'll get into that when we get into components.
The bleach can get it, how does the blood stay?
I'm sorry?
Bleach can go and how does the blood not come out?
It's a special bag that has to breathe because your red cells are still alive, right? I don't know the magic that happens. I just know that we can't bleach them because there's magic that happens. That magical pigtail sticking in. Okay. So the next thing I want to talk to you about is the history. And I don't know if I have this up there now. Let me see what I got. Here we go. Okay, so just a little bit about transfusion history. Where have we been and where are we going? So way back in 1628, William Harvey sliced the vein of a little lamb or whatever, and he noticed that as the heart pumped, blood squirted out, and nothing, eventually the lamb had no blood, right? So that's where he decided that, well, blood must actually circulate through the body and not just go in one direction. Poor little thing gave up his life. And then in 1665, they did dog-to-dog transfusion, found out that if we took blood from a little dog that was almost dead and gave it blood from a bigger dog, that voila, the dog would be licked his wounds and walked away and he was perfectly fine. So they're like, maybe there's something to this. Jean-Baptiste Denis, he did animal to human. I have a video, if you ever want to watch the video, and it goes through all of this, and this is actually quite humorous. Not really, but probably. They had a guy who was psycho, okay? He was beating up his wife, and he was kind of psycho. And they, back then, believed that if you took the blood from a very calm, mild animal, and you put it into the patient who's psycho, that the mildness of the animal would take over and make the person mild and do everything right. So they took this madman, and they gave him blood from a lamb. And of course he had a blood transfusion, right? And he started peeing black urine and almost killed the guy. And they said that he got calm simply because how could he fight? Because he was almost dead. So he actually did recover from this horrible transfusion reaction, only to be killed by his wife a couple months later because she was tired of his abuse. So then it took about 10 years, and finally they said, you know what, this isn't working. Animal to human is not working, so we're gonna not allow it anymore. So they actually made it against the law. So their actually first blood transfusion was in 1795, but nobody published it. So it's kind of like, did the tree fall in the forest if nobody heard it? So they say, right? Okay. In 1818, James Blundell, he was an obstetrician. And, people would go through childbearing, and a lot of women would die from loss of blood. So because the blood of a man is far superior to the blood of a woman, they would take the blood from the husband and give it to the wife, okay? Now, no blood types, right? So he had a device called a gravitator, which was a funnel on a pipe, and he would literally slice the vein of the woman, okay? Put this funnel into her vein, and then put a tourniquet on the man, slice his vein, and he would just simply stand there and squeeze, and it would drip out of his arm into this funnel and go into the woman. And lo and behold, 50% of the time the woman came back to life. Now, if she died because the blood wasn't right? No, she died because she bled in childbirth. It wasn't a blood transfusion that actually killed her. So, yeah. So, anyway, that was his claim to fame. In 1900, Karl Landsteiner discovered the ABO blood group system. So he's considered the father of blood banking. He was actually an immunologist, and he was actually studying the immune system in totally different ways. But he took all of the people that worked in his lab and drew their blood and started mixing it together, the liquid portion and the solid portion of the blood. And what he found is sometimes it stayed smooth, and other times it clumped. And so he actually published his findings, but for about 10 years it kind of went unnoticed until somebody's who was dealing with blood transfusions and trying to figure out a way to make blood transfusions said, oh, well, wait a minute, just because our blood is red does not mean it's all the same. And so then they finally did the first cross match within 7 to 10 years, and lo and behold, when they cross matched the blood first, which meant they took your blood and my blood, mixed it together. If it didn't react on a slide, then we could give you the blood and you would be fine. Okay, so that was kind of the big thing for ABO, was the big voila for ABO, or for blood transfusions. In 1914, we started using anticoagulants so your blood wouldn't clot. Okay, has a lot to do with World War I and how blood was having to go to overseas to help these poor people out. So then we had several different ones, and they're in your paper as far as kind of where they went through. 1927, somewhere between there and 47, they discovered the MNS and the P blood group system. In 1932, they started saying, okay, if we got these people to donate blood, what are we going to do with it until we need it for the next person? So that's when the first blood banks kind of started up. Okay, it's a place to kind of store it. And then in 1940, there was a big race between Landsteiner and Weiner, and they wanted to see who could discover the next blood group the fastest, and they were both discovering things at about the same time. And so they took blood from a rhesus monkey, and they found out that it had an antibody that was very similar to the D antibody that they were finding with moms and babies, and the reaction with moms and babies. And so Weiner actually discovered it, but because Landsteiner was the father of blood banking, they gave him the credit. And so there was a fight over what they were going to name it. And so LW was the Weiner system, and he named it that first, but then it became RH for the rhesus monkey, which was your anti-D. So he got the kudos for that one. But anyway, 1940, Charles Drew. Charles Drew was a black man who did what they call plasma for Britain, because now we're in World War II, and we have guys that are dying overseas because they have a shortage of blood. But it was hard to transport blood products, but if you separated off the plasma from the blood product, you could send the plasma across a boat, right? Because we didn't have airplanes and, fast mail those days. And so he came up, like I said, with this plasma for Britain, and he saved a lot of soldiers' lives by blood transfusions overseas. He was a black man, and he literally died 2 blocks from a hospital because it was a white hospital and they wouldn't allow him in. And he died, and what would have saved him was a blood transfusion. So back in the day, when you had black and white. So the Red Cross now has a Charles Drew program to where they link patients up to donors. So if I'm a patient and I have a leukemia and I'm getting chemotherapy, and so I need blood products every so often, I have a designated donor, so that person would donate blood for me, and it's a 2-year commitment, so that every time as a patient I need blood, I get blood from the same donor. And if you think about it, then I'm not exposed to as many diseases, I'm not exposed to as many antigens, I'm not going to build as many antibodies and all those reactions. So again, that Charles Drew program, so that was kind of neat. And they were looking for people, and they still are. My husband at one time was donating platelets, and he was a good donor for a little boy who had some kind of leukemia. And every time the little boy needed platelets, they'd call up my husband and say, can you come donate platelets? And so he did. I mean, he did for probably 18 months. And then for just some weird, I don't know why, all of a sudden my husband's platelets just bottomed out. And he just had like zilch, and he had petechiae everywhere and red spots, and it was just horrible. So he couldn't donate anymore until he came back to normal, and so that didn't work anymore. But he did for a little while. And it kind of made him feel better. You know, when you're sitting on that machine back in the day, you had a tube in this arm and a tube in that arm, and it's kind of going through a dialysis machine, taking out your platelets. And sometimes it takes 3, 4, 5 hours based on what his platelet count was. But if you know you're doing it for somebody and you're helping somebody, it kind of made the time a little bit better. So. Let's see. 1947, the AABB was formed. In 1950, they started using the plastic bag. You ever watch old episodes of MASH? TV show MASH? And what did their blood come in? Glass bottles, exactly right. So it was actually during the Korean War that they started transporting blood in plastic bags versus because they wouldn't break as much.
Isn't glass a natural coagulant? Like, doesn't blood coagulate in glass?
Well, without an anticoagulant, it will, yeah. Yeah, but again, you have clotting factors in your body that'll kind of get it to trigger too. But then again, it was cheaper to ship in plastic bags, plus you didn't have as much breakage, and so the plastic bag was started to use. Let's see, and then you can kind of read through the rest of those. RhoGAM was 1967. RhoGAM is what we give to Rh negative mothers, so that they don't, have the anti-D reaction. Excuse me. So, I mean, I was born in 63, so 67, that's even my lifetime, so that's kind of interesting. You would hear old stories from grandma or auntie or somebody that said, oh, so-and-so had a baby, and then the second baby was kind of sickly, and then after that, they just kind of kept dying, and she couldn't have any more babies. And again, it's probably an Rh factor, but they just didn't know about it back then, so. In 1970, and I'm not scrolling through here, am I? In 1970, another important one, is we went to an all-volunteer blood donation. So before this, if we needed blood, we would pay you for your blood. And you've heard this, right? Where people say, oh, I'm going to go give blood today because I need money for whatever. And so what they were finding was if you were being paid for your blood was not disease-free, because who was selling their blood? Drug addicts and people living on the street, right? So they weren't exactly clean. They found that people who actually donated their blood, they had less of an incidence of transfusion problems. So that's why in 1970 they went to an all-volunteer blood system where they were not going to pay people for their blood product. And it was pretty strict at first, but if you look around now, I think there is a place where you can actually sell your plasma. And also, if you go to like a blood donation, they'll give you a free t-shirt, or they'll give you a free PSA test, or they'll give you free cholesterol testing, so they're not actually paying you for it, but they are giving you something more than just free cookies and punch, right? So again, something that they've come up with. And then when in 1979 we finally got the blood to last 35 days, which was a pretty big breakthrough. And then, let's see. So in 1983, we got it to 42 days, and that is where we stand right now, 42 days. We have not improved on that one. The blood bag, again, is such that it kind of breathes a little bit. It helps with the pH. It helps with the breakdown and all the other stuff that's going to affect in the bag, and so we've not gotten it any better than 42 days. So which kind of takes you to, if you donate blood and it's going to expire in 42 days, you need to use it. And if you don't use it, at 42 days, blood centers tend to then sell it. So like your reagent red blood cells, where do you think they are? They were actually units of blood that people donated that then expired, and they sold it to a reagent company who then used your cells for no longer good enough for a blood transfusion, but good enough for a reagent. So that's where your reagent cells come from. 1985, we finally started screening blood people for HIV. And so all that blood that people had donated that had either been frozen or had been saved for special transfusion, all of that had to be thrown away because of the HIV. And HIV, is mutated, right? You have HIV 1 and HIV 2, and so we test for both. And then in the 90s, we started hepatitis C screening. 2000, we had the mad cow disease. So then all of a sudden, people who even, pretty much anybody military, been out of the country and been all those places, they couldn't donate anymore. So again, the blood supply got very limited. They've changed that a little bit in that they're releasing it a little bit, not quite as tight. You had to be there for over X amount of time and all that good stuff. Zika virus is kind of big in the news now, but they've been screening for that one and the West Nile virus, they screen for that now. So because they're better at the screening devices, they haven't really had to tell people if you've been in this place, you don't have to go. We just screen the blood for whatever it is that ails you. So it's come a long way. But I do have a movie if anybody wants to watch it. It is pretty interesting. It's kind of gross, but kind of interesting. Ah, da, da, da. All right. Questions so far? On Friday, I'll review and tell you all that what you need to know. Just a lot of information. Okay. So the next one's your genetics. Genetics and immunology really should just be review. You probably did some kind of genetic testing in some other class, maybe a biology class or something in some other classes, so there's nothing new here. But we'll just try to talk to you a little bit about what's important to blood bank. Get my mouse closer. I feel like I'm sitting on the floor. So blood groups. What we worry about with blood groups is on your red blood cells you have antigens. Your red blood cells are not nice, smooth little, bubbles. They're actually little spiny balls, and they have antigens that stick off of them. Okay, and you know from immunology, right? If your body sees an antigen, what does it do? Builds an antibody against it. Correct? Okay, so. Again, antigens and antibodies are what we are worried about in blood bank. So if we have an antigen, do we want the antibody? No. We don't want to give anybody an antibody, right? Okay. So again, we need to know what is it that you have and what are we going to give it to anybody else, and what is the prevalence of having that in the population. So just some genetic terminology. Genetics, again, the study of inheritance, nothing big there. Again, this should just be review. And then gametes are your eggs and your sperm. Your zygote is when they come together, right? Okay. Chromosome, you have double-stranded DNA. So, you know it's in that helix pattern, right? But everything I will talk about with your DNA, I kind of look, have you ever played on monkey bars when you were a kid? I tend to use that description more than, again, being it twisted, but more like a set of monkey bars. So if you took your DNA and untwisted it, it would just have your double-stranded DNA. So when I talk about things, that's probably the reference that you'll have in your head. A gene is your unit of inheritance, just a segment of DNA that holds that recipe for the molecule. Okay, again, a protein. So again, shouldn't be anything too. Gamete, zygote, chromosome, gene, those are a matching on your test as far as the definition and of the matching, so that's all those are. So when you're studying how to study that. Here's your DNA molecule with your bases and all that good stuff, and that's just in case you were ever at a cocktail party and couldn't remember. You can go back to this PowerPoint and refresh your memory. Alright, so on your chromosome, you have a loci, okay, locus. That is on that place on that chromosome where your actual gene is found. Because we have again, think of your twisted chromosome, okay? You have two sides. You can have the gene, and then you have its allele, which is kind of like its partner on the other side, okay? So, if I inherit the K gene from my mother, okay, I've inherited half of my DNA from one parent and half from the other, right? Okay? And so then if my father had the K gene as well, I would inherit two K genes, right? So it's the gene and its allele. the one and the opposite. So if mom had big K and dad had little k, it'd be in your gene and your allele. Okay? Kind of the one. Now this could be the gene and this could be the allele. It doesn't matter. You have one and then you have its allele. Okay? If they are inherited in the double dose state, in other words, if I get two big Ks, I am homozygous, right? Two of the same, homozygous. If I inherit one of one and its allele is different, then they are heterozygous, right? Everybody understands hetero and homo, okay. In blood bank gene studies, we have to worry about dosage. You'll hear me say dosage a lot. Dosage is very important. Because you get one from your mother and one from your father, okay, if they are homozygous, which means if I got two that are exactly the same, I got a double dose of K, right? So my genetic material is big K, right? I got one from mom and one from dad. If I have inherited a gene in the double dose, I'm homozygous, I have a stronger reaction. So again, if you're thinking about your monkey bars, if I was hanging on my monkey bars with one hand, would my grasp be as strong as if I was holding on with two hands? No. So again, double dose, you have a stronger expression. That's going to mean something. Because if you have an antigen, a Cal antigen, and you have a Cal antibody floating by, if I have one antigen to grab onto, then the other one's just hanging there, right? I don't have anything for that other hand to hang on to. I'm not going to have as strong a reaction as if I had two Cal antigens for that antibody to hang on to. Does that make sense? Okay, so again, dosage. Stronger in the homozygous state than it is the heterozygous state. So when we are crossing out our antibodies, when we are eliminating, we can only eliminate in the homozygous state. And when I'm saying that, I mean you got two copies of the exact same gene. You got one from each parent. Heterozygous would mean the reaction was weaker. It doesn't necessarily mean it's negative, right? Just means it's weaker. So we can't eliminate on a heterozygous cell. Okay, all right. But again, we'll get there. Mitosis and meiosis, my cells, my sex cells and mitosis cells, right? Sex cells, female your XX, male your XY. And again, there are some blood group systems that are sex linked. So you're obviously going to get it from the mom and not the dad. Nothing's associated with the Y chromosome. Mendelian principles, this should just be review as well. Mendel basically says that we can either inherit a gene, we can inherit a chromosome. Again, things are just kind of like you throw up a deck of cards and as they fall, you inherit whatever it is that you inherit. Obviously, your brothers and your sisters don't look exactly like you, right? So again, just how things are inherited. In the laboratory for blood bank, we deal with phenotypes and genotypes. What we did in the lab yesterday was a phenotype. We found out what that person's blood type is. Do we know what that person's parents were? No. So the phenotype is what we can actually detect in the laboratory. When we do your blood type, we can tell your phenotype of your blood type, but do I know what your parents are? No. The only way you can find your genotype, your actual genetic makeup, is you have to do a study of the family. We'd have to find out what your brothers and sisters are. Now, maybe based on what your brothers and sisters are, we could tell what your parents were based on what you inherited. But we're not going to find that out in the laboratory. Okay? So phenotype is what we can find in the laboratory. Genotype is your actual genetic makeup, again, based on what your family is. Well, you are right. This paper does feel thick. I don't know what somebody had in the printer. Pedigree chart, in case you've never seen one before. We don't use these in blood bank, but it's just kind of an example where it'll show you if it's male or female, whether they're twins or where they came from. So it's just a standard pedigree chart. In blood bank, okay, we can look at a parent's blood type, and based on what your parents are, we can determine what your children are going to be, or based on looking on what your children are, we can determine what your parents are. So if this was mom, and so the top one is mom, and she tested as an A. Because remember, in the laboratory, that's all we can do, right? We don't know what the family is, we just know that they're an A. But she had 4 children. One child was an A, just like her. Then you had a child that's a B. So what is that saying about the father? The B had to come from somewhere, right? So the B came from dad, right? So then we had a child who's an AB, which means they got one from mom and one from dad. And then we have the O. Where did the O come from? We're heterozygous, right? Not homozygous. We'll do some Punnett squares in just a second, okay? So again, just kind of some fun things that we'll do. So what do? In the laboratory, we do what's called, we're going to jump to population genetics first. It's important for us to know in the population what's the incidence of having a type A person. How do I want to say it? So in Japan, 80% of the population is O, type O. Okay, 80% of the population. So if I'm Japanese and I'm in a car accident and I'm an A, what are the chances of them being able to find the unit of blood in their blood bank for me? Pretty slim, right? Okay, so that's why when we rise to Japan, they had to usually come to the military base to get all those other blood types because all they had really was O. So it's important to us in blood bank to know what are our gene frequencies in our population. Because if I need to give a unit of blood, or say I've got to give a mass transfusion, how many people do I have to go find to find that unit of blood that's going to match them? Okay, so population genetics. Again, by Mendelian laws, the antigen frequencies are determined by the AABB and can differ from race and location. For those of you who are working in a lab, go to your blood bank. You will probably see the chart on the wall. Okay, my chart is on the door in between the classroom and the lab. There's an actual chart that tells you what the gene frequencies are. Okay, again, we don't determine it, it's determined by AABB. But we need to know what the frequency is. Just from what little study I've done, because I go to the lab and I bring usually like 30 specimens for you guys to blood type. I just know that in Belleville we have a lot more A's than we have O's, but in the general population we have more O's than A's. So we must interbreed in Belleville. I don't know exactly why, but we did have more A's than O's. Okay, so we do what's called, for again, population genetics, we do the Hardy-Weinberg equation. I put up there some extra ones just for you to practice. This is on your test, and it's just either an 8 or 10 point question. But it's fairly simple. You have the gene and you have its allele. Okay, so the gene and its allele. So if P is the frequency of one gene and Q is the frequency of its allele, we can determine how many people in the population have that frequency. Which means if I have 100 units of blood in my blood bank, how many units do I have to go grab to find a suitable donor if my person has that gene frequency? My person has an antibody to S. Do I want to give them S positive blood? No. Remember, lock and key, right? I don't want to give you an antibody if you have the antigen. You're going to have a transfusion reaction. So again, P squared plus 2PQ plus Q squared equals 1. So they always equal 1. All right, so P plus Q equals 1. So if my gene frequency for S, so in other words, in my population of 100 people, 30 people have big S. Okay? What are the frequencies of little s? So again, homozygous for big S, heterozygous for big S, little s, and how many people in my population are homozygous for little s, okay? So if I gave you the gene frequency of big S as 0.3, you know that 0.3 plus Q equals 1. So it's a simple subtraction problem, right? 1 minus 3 equals 7. So 0.3 plus 0.7. So 0.3 plus 7 is 1. If you take the square of 0.3 plus 2 times 0.3 and 0.7 plus the square of 0.7. So the square of 0.3 is 0.9. The other one is 0.42, and the square of 0.7 is 49. And again, if you add that all up, you get 1. So what is that telling me? In my population, 9% are big S, big S. Okay? 42% of the population are heterozygous, big S, little s. 49% are little s, little s. So if I have a patient who has an antibody to big S, how many people, if I have 100 units, how many of those units do I have to screen? About half, right? About half. But if my patient has a little s antibody, what are my chances of finding a unit that's little s negative?
Right.
Not very good, right? You have to screen a lot of units to find your units. Okay? So it's important to know. And I will ask you your gene frequencies, and on your component or on your other blood group system, you're going to tell me what they are. Actually, I think I filled them in for you. You're not going to memorize them. You're going to go into the lab, and you're going to find the little chart on their wall, and you're going to say, my person has Kel, big K. How many units do I have to go screen? And you're going to look at the chart on the wall. Okay? But again, you need to be familiar with what the B's are and what the little is. Little e, 98% of the population. So if I have a person with little e antibody, how many of my 100 units are going to be little e negative? Only 2. And we actually found a student a couple years back who was little e negative, and she was in a panic because we'll be able to find any blood for her. Those kind of people, you tend to have to go to the Red Cross because they'll have donors out there that they know what their antigens are, and they'll just call them in and say, hey, we have a patient who needs your blood. Come in and donate. Okay? So again, part of that process. So again, what does it mean? How many units of blood do I have to go grab to find a suitable unit for my donor, or for my patient? So up on the thing, I gave you a worksheet. So again, that's just a one you can play with. You will do this on your test. So again, you're going to know the equation. You're going to know that P, which means the gene, plus its allele equals 1. I have to give you the gene frequency of, the frequency of the gene. So in this case, I give you big R as 0.6. So what is little r then?
0.4.
Right, 0.4. So 0.6 plus 0.4 equals 1, right? So then you just have to plug it into the equation. So r squared, which is 0.6 squared, plus 0.6 times 0.4, plus 0.4 squared, 16, right? What are your answers? Can I click to the answers? So again, 0.4, then you have 4, gives you 1, 6 plus 6 times 4, so 36, 0.36, 0.48, 0.16. Add that all up and you get 1. So what is that telling you? 36% of the population are homozygous for big R. 48% of the population are heterozygous, and 16% of the population are homozygous for little r.
You said this is on a quiz, right?
That's on your first test. Yeah.
Test.
I'm going to give you a gene frequency. You're going to have the formula, and you're going to be able to plug it in. Okay? It's not that hard. Anybody have questions? I think I do have more if you want to play. Okay. So when we know the genotypes of the parents, we have to know the genotypes. If you're an AO, your genotype is AO, which means you know what you actually inherited from your parents. What is an AO going to phenotype in the laboratory? What are we going to be able to tell doing tube typing just like we did yesterday? Okay, so we found out that remember we added the anti-A reagent, right? And if it clumped with that red cell, then we knew they had the A antigen, right? And did anybody here have an O? I think somebody did. It didn't clump with either, right? So you knew it was an O because it didn't have an A and it didn't have a B. Okay. So your phenotypes is what we can test in the laboratory. So phenotypes we can test in the laboratory if you're an A, a B, an AB, which means you would clump in both tubes, your A and your B. And if you didn't clump in either tube, you were an O, right? So that was kind of like detecting it from a lack of a reaction, right? Because an amorph is an absence of a reaction. Because you know your reaction should be that clumping. You didn't get any clumping, then you had an absence of clumping. Okay, an amorph. Genotypes in the ABO blood group system are, in this particular case, AB, BO, AO, or OO. All right, so I'll put up the answer so you can type it into your sheet. Okay, so if I know my parent, again, as their genotype is an AO, which means they inherited an A from their mom and the O from their dad. The other parent is a BO. So a B from one parent, the O from another parent. Okay? Now they give half of their genetic material. Right? You have the egg and the sperm, they split in half, you get half of their genetic material. So if one parent was an AO and the other parent was a BO, and they had 4 children, one child is going to be an AB. Right? One from mom and one from dad. AB. Woohoo, got a little bit of mom and a little bit of dad. A little bit of both of us. Okay? The other child would be a BO. How are they going to type in the laboratory? When we do the tube typing like we did yesterday, what reactions are we going to get? Nothing in the A tube, right?
Mm-hmm.
Clumping in the B tube. How do we know it's an O?
If we got a little bit.
We don't know, right? We don't know. Only because we found out from the genetic history. Okay, we can't test for that in the laboratory. Okay. Then if my other parent, again, was a BO, then the third child would be an AO. And they're going to test in the laboratory as an A. Then the last child is going to be an OO. So you have an A mom and a B dad. And you have a fight going on in the upstairs OB ward, right? Because how can that be? Must be the mailman. Doesn't have my blood type and doesn't have your blood type. We get this call at least twice a year. Because mom and dad are about ready to kill each other. So we go upstairs and we do draw the Punnett square and we say, well, we now know what your genotype is. Because we can determine. I'm not saying it's not the mailman because we still have to go a little bit farther. But it could possibly be your child. Okay? Based on what you have inherited. So I gave you some other ones. So again, if in the laboratory we did a blood type on a parent and the A tube agglutinated and the B tube agglutinated, we would say that parent was an AB. We know their genotype, right? And let's say the other parent typed as an O. The A tube was negative, the B tube was negative. They were an AO. So if you had an AB parent and an OO parent, what would their children be? AO. If you're going across, it'd be AO and then and then and BO, right? Okay. Now if you had a homozygous B And a homozygous A, what are their children going to be? All of them are going to be AB, right? That's all you have to give. Okay? So on the next one down here, you have one parent who's an AB. Can you have an O child? No, you cannot. So an O parent cannot have an AB child, right? Because they don't have an A or a B to give up. So an O parent can have an A or B child. And an AB parent cannot have an O child. Correct? So those are just filled in. Any questions? So on your next test, you are going to draw a Punnett square. I'm going to give you the genotype of the parents. Okay, I'm going to give you how to fill in the top square and the side squares. You are then going to take and match your gene up and tell me what the children are. Okay? Of that very first box, if that were the test question, and I told you one parent was AB, the other parent was an O, fill in the Punnett square. You would fill it in just like that, right? What are the what are the phenotypes of the children? Phenotypes of the children. A or B. Does everybody understand that? What can you test in the laboratory? What are the genotypes of the children? AO
and BO.
and BO. Does everybody understand that? Dominant and recessive, just a little bit about dominant and recessive as far as what you're inheriting, whether or not your children are going to have red hair or whether they're going to have blue eyes or all that good stuff. What's important here, in blood group systems, it is codominant, which means A does not overshadow B. If you get A from dad and B from mom, you're going to find both in that tube of blood. They are not going to overshadow each other. There is no dominant, there is no recessive. Everything is codominant. Okay? Remember, O is an amorph, which means it has no detectable product. Not that it's recessive, it just does not have a detectable product. Okay? So all blood group systems fit in that codominant pattern. Autosomal recessive equal between the sexes. There are some blood group systems that are linked to the X chromosome, okay, the X gene. So again, male to male can't pass that on, right? Because for a dad to pass it to his son, that's the Y chromosome. So female to male, mom sends her X chromosome to her son, or again, female to female, which again is just like with the hemophilias, which you probably learned about it away. So paternity testing. We can use blood types to determine whether or not that's the parents of that child. Now they do maternity testing, but usually that's very rare because usually we know who the mother is, right? We know who's about. Usually they'll do maternity testing if they find a baby in the trash can or something like that, but usually maternity testing is not done around. So again, they use blood group antigens and HLA antigens as genetic markers to find out whether or not this is your child. Okay. You have your direct exclusion, which means if the child has a marker that neither parent has. Can't be dad, right? Indirect exclusion, if the child lacks a marker they should have inherited. So again, if dad is CC and the child did not get the little C, then we know it can't be their child. Okay, so again, direct or indirect. So far we've talked about the ABO blood group system, which will be your lecture next week, ABO and RH. So ABO is the most important blood group system. So we can do a simple ABO and we can do an exclusion, because if my child is an AB and dad tests as an O, we know it's not his kid, right? Okay. So again, we can do a simple thing with blood testing. But you're going to learn about a whole lot of blood groups, okay? Because after the ABO, you've got the RH, and then we learn the Lewis, and then the MNS, and then the P, and then the Lutheran, and we go on and on and on. Okay. Of course, you guys are going to tell me all those. I'm not going to tell you all those. So we can use a lot. The MNS blood group system, because there's so many antigens in the MNS blood group system itself, is usually the one that they use for paternity testing, because that's the one we can usually rule in and rule out, that having to go to a lot of the other blood group systems. A lot of it has to do with whether or not it's present at birth, right? Because do I want to wait till this baby's 4 years old before I find out whether or not that's the father? No. So again, some of them, in the ABO blood group system, it is present at birth. You do have a blood type when you're born, but not all of them. Okay, so again, you're going to be learning that as well. And the MNS blood group system, you do have them when you're born. So it would be easy to do paternity testing. It used to be that we had to wait until the kids were about six months old, but now we've gotten even better, so we can do it pretty much from cord blood. So it has gotten a little bit better. Dun, dun, dun.
Can you do it when they get older?
Do what, I'm sorry?
Can you do it when they get like probably my age or something?
Oh, absolutely. Then it gets easier.
Really?
Oh, yeah, because not all your antigens are present at birth. So how are we going to know if you got it from dad if it's not on you until you're 2 or 4 years old?
Mm-hmm.
So we know some are present at birth and some aren't. So those are the ones we're going to use for paternity testing. So when we get to antigen typing, you're gonna have you guys draw your blood, and so then we're gonna find out what antigens you actually have on your blood type. So you're not just an A or a B or an O. It could be an M or an N or an S or a Lewis or a Lutheran or a P. I feel sorry for the P people. Because if you had the antibody to P, you urinate blood when you're cold. So if you guys don't have that problem, then you know you don't have that problem. So you feel for that little guy that's out there, turning the stop and go sign at the construction site. That wouldn't be a good job for him. To learn about all those.
Mm-hmm.
I'm sorry?
I said it's funnier because you called them the P people.
The blood group
And
is
then
P.
you said that they P blood.
Yeah. It's an easy one to remember because the blood group is P
Yeah.
and they have paroxysmal cold hemoglobinuria, which means they pee blood when they're cold. So it is an easy one to remember. It is an
Is
easy one
that
to
bad
remember.
for them or?
I'm sorry?
Is that bad for them, right?
Well, yeah, it's not
They're
good.
gonna lose
They
blood.
can become they
Okay,
don't want
so
that
how-
job.
So how would they get like transfusions or something like that if they wanted like?
Well, we transfuse it at warm temperature, right?
Oh.
We don't give them cold blood.
Okay.
You pre-warm it
Oh, okay.
and not in the microwave, as we will learn. Now, the nurses are supposed to leave them out. The magic number is 30 minutes. Blood can be out for 30 minutes, and if you don't transfuse it, you can bring it back to the blood bank. If it's over 30 minutes old, you have to dispose of it. So their number is 20, so they can leave it out at 20 minutes. But yeah, we have had a nurse that put it in the microwave to try to warm it because they wanted to go to lunch. You know what happens to a bag of blood that you put in the microwave?
That sounds like an awful idea.
Yeah, cooks it just like your hamburger when you're thawing it because you want to make your spaghetti sauce real fast, right? Yeah, they're not a nurse there anymore. The blood bank is very interesting. It is very interesting. The labs are very repetitious. You do the same thing over and over and over again. I will stress to you over and over and over again that especially as a new tech, if you don't understand, you've got to ask. That's where they get into trouble. It's not that they've done something wrong, it's that they didn't ask about what was going on. So I know I ran through that fast, but again, that should be a review. Friday will be immunology, which should just be a review, because that's what you just learned in serology. In serology, you did IgG, IgM, IgA, D, and E, correct? In blood bank, we only deal with G's and M's, okay? We will talk a little bit about A's when we get to transfusion reactions, but that's like week 6, so we're going to go away. So we're going to be dealing with M's and G's. So if you do not remember the difference between your G's and your M's, you need to review that. Yes?
IgE is when
Yes, but these aren't allergic reactions. These are antigen antibody reactions, not allergic reactions.
So an allergic reaction, somebody can't have.
With your IgA, you can have an allergic type reaction. We actually call them anaphylactic reactions with your IgA. But we deal with G's and M's. So if you don't remember the difference between your G's and M's, you need to review that, because that's a big part of your test as well. On your test, it'll be on Monday. I give you a characteristic, and then you're going to tell me is it G's or M's or both, or none. Okay, so we'll probably fly through that one as well. I'm sorry? About whether or not they react with complement, or whether or not they're present at birth, or whether or not they're
placenta and the other one's not going across the placenta.
right. So again,
But that's why I'm not getting happy. Okay, well, I guess we get it later.
yep, you don't remember. Now, certain enzymes will destroy certain ones. So blood
That's
bank
how I
is
remember
fun.
G. That the IgG is a G because he's going across the placenta just with no problem.
Well, there you go. However you want to
And
remember
then
it.
the other one is
Any other questions on anything we raced through today? You need me to do another Hardy-Weinberg equation? You need me to do another Punnett square?
Can you do another equation?
Sure, sure. So let's see. Give me one that's easy to tell a big from a little when you're writing it. Okay, B.
Six
How about
simple.
B and little b? The gene and its allele. All right. So if my gene frequency of B is 0.2, what is my gene frequency of my little b?
Same?
it's 0.8.
Okay, how did you get that? Because it always equals 1. So if one is 0.2, the other one must be 0.8, right? Okay? So the equation is the first one squared plus 2 times the first and the second, right? And the third one squared. So 0.2 squared plus 2.2 times 0.8 equal, oh, I'm sorry. Plus 0.8 squared. Is your equation correct? So far so good? So far so good? Okay, so 2 squared is 24. 0.2 times 0.8 times 2. 32.
That's what that is.
So 0.32. 8 squared is? 8 times 8 is?
Oh, sorry.
64. And again, that equals 1. So did we do our math right? No, we didn't do our math right. Did we? Yeah, we did. Yeah, we did. So that equals 1, right?
Yes.
Okay. So in my population, how many of my patients are VP? I said percent. This should be 0.04, right?
Yeah. Yeah.
Zero four?
Yeah, it's your.
I bought my grandma and mother's home, so it was like.